I'd like to welcome you all here today. My name is David Bowe. I'm from the Walter and Eliza Hall Institute, and it's my distinct pleasure to introduce Professor Roberts. Andrew Roberts is, and he's got a very long title here. I don't know how he would ever fit it on a card, but Andrew Roberts is the Metcalf Chair of Leukemia Research at the University of Melbourne, a clinical haematologist at Integrated Haematology Department, Royal Melbourne Hospital and Peter MacCallum Cancer Centre, the head of clinical translation at the Walter and Eliza Hall Institute, and the research and education lead for haematology across the Victorian Comprehensive Cancer Centre. And I couldn't do that in one breath. Now, I've known Andrew for a very long time, and I'll just share with you some material that I plagiarised from a letter that was written by Mark Rosenthal, who was who's also one of the directors of research here at the VCCC. And this was to mark the publication by Andrew of a paper in the New England Medical Journal, which is the world's most prestigious medical journal. And so Mark wrote, "I would hazard a guess that Andrew Roberts has achieved something that very few individuals in the world have achieved, a significant and direct involvement in a basic and translational program that evolved into leading the early phase clinical trial of a new drug that almost certainly will vastly improve the care of patients with cancer." And he goes on, "I've been around long enough to believe that it is one of the greatest ever achievements from this campus in any field of medicine. And what a story, arising from work in the 1980s through a succession of basic and translational discoveries and then to this clinical trial." And if you read the paper in the New England Medical Journal, it's interesting because of the illustration of the number of teams that have played a role in this work. So the addresses refer to the Royal Melbourne Hospital Department of Clinical Haematology, Royal Melbourne Hospital Bone Marrow Transplant Unit, Royal Melbourne Hospital and Walter and Eliza Hall Institute of Medical Research, the Victorian Comprehensive Cancer Centre, University of Melbourne, Peter MacCallum Cancer Centre. And so they're just the ones right from this campus here. But there's also the Dana-Farber Cancer Institute, the Memorial Sloan Kettering Institute, the University of Texas MD Anderson Cancer Centre. And these are the biggest cancer research institutes on the planet. And this was all led by Andrew Roberts. So take it away, Andrew.
Well, thank you, David, for delivering my talk. I'll flesh it out a little bit. That's a very gracious introduction. What I'd like to do today is to talk about a new drug that owes its origins and owes its clinical development and owes where it is today to science that originated here in Melbourne, to clinical trials that were conducted here in Melbourne, and to the vision of people over several decades here in Melbourne, working in an absolutely crucial and necessary partnership with industry to get us to where we are today. Before actually talking about the drug, I did want to make a number of disclosures. I've actually got more things to disclose than I have jobs, and you heard the long list of jobs that David attributes to me. But we are talking about a medicine, and medicines are sold for profit. They're sold for the benefit of people with disease, but they're sold for profit, and any time profit's involved, there's potential for conflict of interest. So this is a list of my associations with the pharmaceutical industry, and the key one I want to point out to you is the fact that one of my employers, the Walter and Eliza Hall Institute, receives milestone and royalty payments related to the drug I'm going to talk about, Venetoclax, from the two pharmaceutical companies that we co-developed the drug with. The other thing I'm going to say is some of what I'm going to talk about is the use of Venetoclax in people who have previously treated chronic lymphocytic leukemia. And in that circumstance, I'm talking about a place where the drug's been approved for use routinely in Australia by the Therapeutic Goods Administration. But much of the other that I'll be talking about is still in the research phase, so isn't part of standard clinical care as yet. And the final thing I'm say is I wear many hats, but I'm actually talking to you today as an academic, not as an agent for pharma or indeed any of my particular employers. I am hoping that what I am talking about today will provide an illustration of what the VCCC is going to continue to be able to achieve in the years ahead. I want to reflect on why my career has been about trying to think about new treatments for cancer. And one of the key reasons for that is because, in fact, that's how you go about making a big difference for people where our current treatments aren't delivering the goods. When we think about how to make big differences for cancer outcomes, not just for individuals, but for large numbers of people, basically there's two ways you can do it. One is you can prevent the cancers happening in the first place, and the other is, if the cancer occurs, then have a treatment that works better than we currently have. Whichever way you choose to approach it, and it will be different for the different types of cancers, it's essential that this is all based on research. In fact, every single advance in the control of cancer over the last 100 years has been the product of research, not luck. If we want to think about prevention as the best way about going about making a big difference in terms of controlling cancer, we need go no further than look at the impact of tobacco control. And the graphs I show you here on the right-hand side show what was, in the blue, a rising rate of lung cancer in men, and what below that is a still rising rate of lung cancer in women. And then suddenly that blue line starts to come down, and indeed the pink line starts to flatten out. And the reason for that is introduction of controls over tobacco, and the rates of smoking have fallen off very dramatically and fully explain the reduction in the number of lung cancer diagnoses being experienced in the community. I'd love to be able to say that lung cancer treatment is a whole lot better than it was 20 years ago. It is, but the difference that those improvements have made have been quite small. And you can tell that by looking at this these curves on the right-hand side, where you see that still for people diagnosed with lung cancer, the majority of people are going to die of lung cancer. If we had a better treatment, you'd see that a much greater proportion of people would still be alive, even if they were diagnosed with lung cancer. So beating lung cancer, stop smoking. Reduce that rate in the community, you'll make a big difference. And that's triumphant research that was done in the 50s, implemented in the 1980s and 90s, and is reaping benefits now. What about drugs? I want to give you two examples that have inspired me during my career. The first is a drug called rituximab, which has revolutionised how we go about treating patients with the most common types of lymphoma. Unfortunately, lymphoma is increasing in incidence in our community. In other words, it's becoming more common. And you can tell that by these curves on the right-hand side, which are going up, particularly the ones at the top. That's the incidence of the disease in men and women. If you look at the curves below that, that's the death rate in men and women. And you see it's going up and then starting to come down. And that reduction in death rate isn't due to the development of a new chemotherapy drug. It's development of a new drug called rituximab, which was one of the first two monoclonal antibodies introduced into cancer medicine. And if you look at this curve on the left-hand side, you'll see that there's a dramatic decline in the number of deaths, the chance of dying from the disease since the introduction of that drug. So early in my career, I saw what rituximab did to the outcomes for my patients, and I recognised just how powerful the introduction of a new drug could be. Soon after, there was another new drug, also in the blood cancers, and as a hematologist, that's the sort of cancers I look at. This was imatinib, or for chronic myeloid leukemia. This is actually a tablet, a new drug that turned a fatal disease, uniformly fatal disease, into one which can now be cured with these tablets. And that's a tremendous outcome. And again, you'll see the dramatic effect it's had on the chances of dying from this disease through the introduction of a new therapy. Both these examples of new treatment have something in common, and that is that they're targeted. And new modern cancer therapies are now all targeted. Why? Because we want to have a preferentially negative effect on the cancer cell over the normal cells in our body. How do we do that? Well, one way is to understand the multiple abnormalities that occur in the genetic makeup of the cancer cell that lead it to behave as a cancer. We know that for essentially all cancers, this requires multiple changes, and we've been able to exploit this in certain circumstances to work out the Achilles heel of cancers that occur when there's a specific genetic change. And today's talk will be about how we've gone about following this approach to come up with the Achilles heel for a disease called chronic lymphocytic leukemia. What that means, though, is you have to understand the genetic changes, you have to understand what they mean in the disease before you can come up with the treatment. And again, this is all about research. The disease I'm going to emphasise the most today is one called chronic lymphocytic leukemia. It's the most common blood cancer it's the most common leukemia in our society. It's typically not a disease of kids, it's a disease of older adults, and many people will actually end up being diagnosed and never needing any treatment. However, the majority of people will need some treatment, and for some of these people, treatment is unsatisfactory, and chronic lymphocytic leukemia will lead to their premature death. This is the 20% of patients I have on the slide with a very poor prognosis. And it's these people that we're looking to develop a new treatment for. Now, why were we doing that? Well, we knew that chronic lymphocytic leukemia cells are abnormally long-lived and that they accumulate in the body so that the blood film is full of these little blue cells, which they're only blue because of the stain that's been applied, so we can see them under the microscope. They're actually white blood cells. They're all mature lymphocytes. They're all malignant. And instead of dying as they should in the body, they're building up over time and beginning to cause problems. We now know that the reason that they live for so long is because they have too much of a protein in them, and that protein is called BCL2. The reason they have too much BCL2 protein is because of several different genetic changes which ultimately lead to too much BCL2. So genetic change, too much BCL2, cells accumulate, cause problems for people. And the function of BCL2 and its role in cancer was first discovered at the Walter and Eliza Hall Institute here in Parkville in 1988. Well, that was when it was reported. It was actually discovered the year earlier by this man who actually introduced me, David Vogt, when he was a young PhD student working with Suzanne Corey and Jerry Adams at the Walter and Eliza Hall Institute. Now, David began his research on this gene BCL2 at a time when no one really knew what it did. It had been discovered in a different type of lymphoma, and one we'll come back to, one called follicular lymphoma, as something that was regularly seen. So people knew it must be important, but exactly how it led to cancer was not clear. And the great discovery that David made that was published in the prestigious journal Nature was that too much BCL2 meant that cells could live when they should die, and so that they would accumulate over time. And that's a very favourable scenario if you want to become a cancer. It didn't take people long after David's discovery to then start thinking how you could exploit that knowledge. And if you think about what I've just said. Too much BCL2 means the cancer cell will live. Could you think of a drug that would turn the BCL2 off? And wouldn't you expect then the cancer cells to die? So the first patent that claimed to be able to do that was in fact published the year after David's initial discovery in 1989. People coming up with a strategy to target BCL2 to lead to cancer cells to die. Unfortunately, that strategy ultimately failed, and several others have been tried as well. And so it turned out that as good an idea as this appeared to be, it was going to be very difficult. It was very difficult for a number of reasons. Firstly, BCL2 is a bit of a moving target. It's not easy to get to. Secondly, people were overly simplistic about how they went about it and didn't use rigorous science as the basis of trying to develop their therapies. Thirdly, people didn't really understand the consequences of BCL2 in both normal cells and in cancer cells fully, and they didn't understand the interaction of BCL2 with other proteins in the cell that ultimately needed to be understood before it could be targeted. So this led to 15 years of frustration in the field. Ultimately, we were able to develop a drug that did specifically target BCL2, and as I'll show you, did cause cancer cells to die. And this was made possible through critical understanding of the biology of BCL2 within cells, and then the tremendously influential breakthrough work in drug design by scientists at the pharmaceutical company Abbott Laboratories, now called AbbVie. Just to take you through the scientific discoveries that have paved the way, I've talked to you about the discovery of the abnormal gene BCL2 in 1984, then David Vaux's discovery in 1988, and then I've alluded to you the large amount of basic research that was performed between 1988 and 2005 to get us to the point where we're poised to make the development of a new drug. And I want to emphasise that Melbourne scientists led the field. They weren't the only people in the field, but they absolutely led the field along with colleagues in the US and Europe. And so by the early 2000s, there was a very good regulation, good understanding of how this BCL2 protein was regulating whether cells would live or die. And here I want to emphasise regulating whether normal cells would live or die or cancer cells. And ultimately, it's due to the interaction of the BCL2 protein with several other proteins in the cell. And I've oversimplified it here, but I also have included some of the detail in the boxes. The details are not particularly important, except to say there are other proteins that function just like BCL2 in cells, and we had to understand what they were about and whether they would subsume the function of BCL2 if it was targeted. Secondly, there's a family of proteins called BH3 only proteins, and they're shown here, this BH3 in red. There's a number of those, and their job is to bind to BCL2 in the cell and switch it off. And it was understanding these BH3 proteins and what they did that gave a clue to how the drug could be developed. And finally, there's these Bax and Bac proteins down the bottom in purple. They're the actual proteins that will cause a cell to die. And in healthy cells, as I'll show you in a minute, BCL2 stops those proteins working. So we end up with a very complex system that had defied understanding for 15 years, but ultimately science was in a position to be able to explain why a cell lived or died. Now, of course, most normal cells want to keep living and are happy. Normal cells will die when they've come to the right age, or when they come under a stress. And I've listed some stresses there under cellular stress. Cancer cells are susceptible to the same stresses. In fact, more often than not, they're more susceptible to these same stresses. And one of the stresses that we can give a cancer cell is to give it chemotherapy or radiation. So I want you to bear that in mind. So the idea to target BCL2 with drugs was to come up with some drugs called BH3 mimetics that would mirror the way these BH3 only proteins acted within the cancer cells. So the cartoon on the far right there, you'll see a normal cell with some BCL2 there. It's happy, it's living, it's not undergoing apoptosis. And apoptosis is the word for cell death that we use here. When that same cell undergoes stress, these BH3 only proteins suddenly become active. They interact physically with BCL2, they switch it off, and because BCL2 switched off, the cells will die. What happens in a cancer cell here, I'm talking about a blood cancer cell, well, there's too much BCL2. So the same amount of BH3 only protein that's come up because of stress is now no longer strong enough to overcome the barrier of BCL2, so the cancer cell lives. So the idea of the drug is to have a small molecule, a tablet that people could swallow, that would get into the cancer cells, would go to bind BCL2 and act like these red BH3 only proteins, switch off the BCL2, and then allow the cancer cell to die. And the first drug that really did this was a drug called ABT737. It was produced by Abbott Laboratories. It was a product of understanding the biology that had been defined in Melbourne and elsewhere with their revolutionary pharmaceutical drug design. Subsequently, that drug, which was only good for use in the test tube, was turned into a drug that could be given to patients, and that drug was called Venetoclax. And I'm pleased to be able to say that some of the earliest trials with Venetoclax were conducted here in Melbourne, and I'll touch on that in a moment. Before that, though, we were able to collaborate with Abbott and test these drugs in the test tube and see just how active they were. And this was a really pivotal part of the process that first brought academia and pharmaceutical industry into a collaboration. That led to a series of events, and I'm not going to go through these in detail, but firstly, here in Melbourne, we were able to prove that the drug worked as it said it would. This was a fundamental flaw of one of the earlier drugs that had tried to be developed for to target BCL2, but in this instance, the drug designed by Abbott did what it said it would do. Work of PhD students, particularly Kylie Mason and Andrew Way, led us to identify the diseases that we might want to use such drugs in when we came to clinical trials, and that's chronic lymphocytic leukemia and some of the B-cell lymphomas. In the laboratory, using models of disease, we were able to identify that we'd have a problem with the first drug because not only did it target BCL2, but it targeted one of the other proteins in the cell that could also keep cells alive, called BCLXL. And the consequence of targeting both of those was that not only would the lymphoid cancer cell die, but also some of the normal cells, particularly the blood platelets. We thought this might or might not be a problem. We went on to conduct a phase 1 clinical trial with Venetoclax here in Melbourne at Royal Melbourne Hospital and Peter MacCallum Cancer Centre, and John Seymour was the lead investigator here at Peter Mac on that clinical trial. And together with international sites, and David Vo read out several of those, we were able to show that this first generation of inhibitor of BCL2 was actually active in about 1/3 of people with chronic lymphocytic leukemia. But unfortunately, that side effect that we predicted in the laboratory turned out to be true, and that many of these patients also had falls in their platelet count. Now that didn't cause any major problems directly for the patients, but did mean it was a bit hard to go on with this drug. We were also able to treat different types of blood cancers. We're combining the Venetoclax with that monoclonal antibody that I talked about at the beginning, rituximab, and we were able to show that the two classes of drug could work together for additive benefit. But what we really learned from the first clinical trials was that if you wanted to target BCL2, you should target only BCL2 and not any of these other proteins, and that way you might be able to get a stronger effect where the benefit for most patients and fewer side effects. The other thing we learned was that this class of drug, when given to people, did lead to specific cell death of leukemia cells. And this slide shows you close-up pictures of blood from patients who are on the clinical trial. The blood cells at the top of each of those two boxes are normal blood cells, and they're looking very healthy. The blood cells that are arrowed are chronic lymphocytic leukemia cells, and they're actively in the process of dying as a direct consequence of the drug. So we knew that you could target the CLL without affecting some of these other white blood cells. So the next few years was about rising to the challenging of designing a drug that was a bit like these previous drugs, but it only targeted BCL2 and not BCL2 and BCL-XL. This was done in a commercial partnership between Genentech, AbbVie, and the Walter and Eliza Hall Institute, and it led to the development of a drug called ABT199, which we now call Venetoclax. And this slide summarizes for you the fact that this drug only interacts with BCL2, only inhibits it, and did not interact in any physical way with BCL-XL. At least that's what the laboratory said. That's what worked in the test tube. Kylie Mason, while she was doing a PhD, and Seong Lin Kaur, a pediatric oncologist from the Kids Hospital, who was also doing a PhD with myself and David Wang at the time, went on to test whether this was true in normal cells and cancer cells from patients with chronic lymphocytic leukemia. And we were able to show that the new drug Venetoclax specifically killed the CLL cells at least as well as the previous drug, in fact twice as well, but it did not cause the platelets to die. And so we had a lot of confidence taking this drug into clinical trials. And the first clinical trials of this drug were initiated here in Melbourne. The first patient treated at the Royal Melbourne Hospital. The first 3 patients treated here in Parkville at Royal Melbourne Hospital and at Peter MacCallum, again with John Seymour as a very close collaborator in this work. And something pretty remarkable happened. Anytime a person's asked, are they interested in going on a phase 1 study, so that's at the very early part of drug development, we're asking people to have a lot of faith in the medical system, to trust us that this might be a good idea, because in fact we're asking people to take drug that we really don't know too much about. The very first trial is called a first-in-human trial, and this was a first-in-human trial. So the very brave people with chronic lymphocytic leukemia who went on this trial had no other treatment options. They weren't promised that the drug would work. What they were told was that we've got a new drug, we don't know what dose to give, we don't know how often to give it, we're trying to work that out, will you help us? And they put their hands up. I can tell you that we knew immediately that the drug was likely to be beneficial, because in each of the first 3 patients there was a dramatic reduction in the amount of chronic lymphocytic leukemia present in the blood after they took a single tablet. And this graph on the right-hand side in red shows you how quickly the leukemia cell count dropped within 8 hours of taking the first tablet. The blue graph shows how quickly a breakdown product of the leukemia cells called LDH accumulated in the system. And what we at Royal Melbourne and John here at Peter Mac observed was these people were having what we called biochemical tumour lysis, which means that the leukemia was breaking down in front of our eyes, melting away, if you like. That actually meant that we were giving too big a dose as the first dose. We certainly want to shrink everything down, but you don't want to have this tumour lysis. So we had to work out a way to give the drug safely, and we were able to do that. This drug did melt away chronic lymphocytic leukemia in the majority of patients. This is a slide of a patient of my colleague Mary Ann Anderson and I at Royal Melbourne Hospital. This chap had very bad chronic lymphocytic leukemia that didn't respond to any of our standard treatments anymore, and he had very large lymph glands in the neck, and within 6 weeks of treatment he was, as he said, a new man, and those lymph glands had disappeared. The trial is still ongoing. It started in 2011. We still have patients taking this medicine. Overall, as I'll show you, it's worked in about 79% of patients. This slide is just an illustration of how much reduction in the leukemia we were able to see in the patients. Each bar is an individual patient. They're colour-coded because we didn't know the right dose, so different people got different doses. And the leukemia is present in the blood, in the lymph glands, and the bone marrow. So there's 3 different graphs for all the patients. These are what's called waterfall plots. The idea is that you start with a baseline amount of disease. If the disease is getting worse, they go up. If things are getting better, they go down. And the only thing I want you to take away from is the vast majority of people, they went down, and they went down by a large amount, including to 100% resolved in a minority of patients. We can tabulate these sort of results, and it's shown here. Overall, 79% of the 116 patients who went on this first-in-human study with chronic lymphocytic leukemia had a response. Overall, 1 in 5 people, in fact, had what we call a complete response, which is where you don't see any leukemia on blood counts, CT scans, or bone marrow biopsies. And this drug seemed to work just as well whether you had some of the nastiest genetic changes or whether you were frankly refractory or did not respond to our best other treatments at the time, fludarabine. So here's a drug that was active against chronic lymphocytic leukemia. It killed the cells by persuading them to kill themselves, undergo apoptosis, and it did so regardless of the genetic changes within the cell or whether they were still susceptible or resistant to standard chemotherapy. So what we learned from this first-in-human trial is listed on the slide here. We knew that if you had a drug that inhibited BCL2, it would be very active because it would kill CLL cells, it would kill them by this process of apoptosis, and it would kill them independent of specific genetic changes, including loss of p53 function. And what's more, worked throughout the body. We even saw some patients where using our most sensitive tests, we couldn't see any sign of the disease. Now, I want to stress that giving this drug did not cure any of these patients. Some of these patients are still in remission and doing very well, but I don't believe that this drug will have got rid of all the leukemia cells in these patients, or if it has, it will only be in a very small percentage of them. We were able to work out the right dose and the safest way to give the drug, and that's how it's been given ever since. Very soon after, we followed up our participation here in Melbourne with an international phase 2 trial in patients with the worst forms of chronic lymphocytic leukemia, those with deletion 17p as a chromosomal abnormality, which means that they lose their p53 function. This was led by a colleague in Germany, Stefan Stilgenbauer. And that trial, which had very rigorous controls on how people were assessed, confirmed an overall response rate of 79%, and that some people went into remission In other words, it recapitulated what we'd seen in the first in-human trial. Furthermore, it showed that many of these patients were getting ongoing benefit, as shown in these Kaplan-Meier plots, which I won't go into detail now, but suffice to say that many of these responses are sustained. Collectively, those data led to the approval of this drug for use in the United States by the FDA in April of last year, and by the TGA here in Australia in January of this year. And the drug is now on the market for patients with certain types of bad chronic lymphocytic leukemia. And I've summarised the findings as we've gone along. But there are other blood cancers where there's too much BCL2 in the cancer cells. And I've listed some, but not all of them here. What I want to show is that if BCL2 is the target, then you want to be sure that the disease you're trying to treat has BCL2 in it. So these diseases have been selected because many patients, the cancers in many patients will have too much BCL2. In CLL, essentially every cell in every patient has too much BCL2, and I want you to keep that in the back of your mind, because that's not quite true for these other ones. Those in the red rectangle are forms of lymphoma, and I want to touch on the results that we've seen still in the research setting with this before touching on the others. This is another so-called waterfall plot, and you'll remember going down is good, going up means the disease is getting worse, and you'll see a bit more of a mixed bag here. The different colours don't represent different doses. In fact, we've lumped all the doses in together. They represent different types of lymphoma. But what you can see, that the ones on the farthest right, mantle cell lymphoma, the majority of people respond. Similarly, about 50% of people are responding there with follicular lymphoma, but it doesn't look quite as good for this, these dark blue bars, which is the most common type of lymphoma, an aggressive form called diffuse large B-cell lymphoma. But even there, some people are responding well. And this slide just summarises for you the overall response rates, or what proportion of patients had a good response to the drug. And I've kept the results for chronic lymphocytic leukemia up there as a reference point. As you look along, you see follicular lymphoma, about 80% of follicular lymphoma has a lot of BCL2. About a third of patients respond, and only about 14% of people get what's called a complete response. That's what a CR is. That's when we see no sign of the lymphoma. And as you look down, you'll see that the best results other than CLL are in this disease, mantle cell lymphoma, where the results are quite interesting. Two other diseases where BCL2 may be there in high abundance in at least some people with these cancers are myeloma, a nasty disease of the bone marrow, bones, kidney and blood, and acute myeloid leukemia, a very aggressive disease, most common in older Australians, that replaces the bone marrow, and unless people respond well to chemotherapy, has a very poor outcome. And in both these diseases, we're seeing that some patients will respond to this Venetoclax on their own, and indeed some patients achieve a complete response. One thing I want you to take away from the last two slides is that I think there's a big difference between a disease where most people will respond and some people will respond very well, and diseases where it's a bit of a mixed bag, only a minority of people will respond and very few will have a complete response. Because what that says is you need to be thinking about how you can combine this drug with other drugs so you can get better outcomes for your patients. And so that brings me to this part of the talk, which is really about how do we build on what we've learned with the BCL2 inhibitor Venetoclax to date. And here's this slide that I showed you earlier with the BH3 mimetic drug, Venetoclax, coming in from the left-hand side, filling up the VCL2. If we want to kill more cancer cells, and we've inhibited VCL2, and they're still alive, it seems pretty obvious that the way you can add to that is come with a different stress that will kill the cancer cell in a different way. And that combined with the BCL2 inhibitor, we're likely to get a better outcome. And so the challenge in research has been, which diseases would we do that in? My suggestion is we start with the diseases where we saw the Venetoclax have some activity, but not as good as CLL, and which drugs to add. And my take-home message about that is fit for purpose. So we choose drugs that are active in their own right against the specific diseases. What are we aiming to achieve? If we're using BCL2 inhibitors in combination with other drugs, well, that's pretty obvious, and we want to do better for our patients. So, but what does that mean? I think that means for people where we think Venetoclax would do something on its own, we actually want to get everyone to respond and everyone to stay responded, and in fact we want to get rid of the disease, we want to cure them. Where the diseases are only partially sensitive, we want to really ramp up the response rate, we want to increase the duration of the responses, and we want to see if we can actually stop taking this medicine, because one of the problems with many targeted medicines these days is that they're ongoing treatments for years, rather than old-fashioned chemotherapy, which was given for weeks or months and then you stop. With these targeted treatments, the standard has been start them, stay on them as long as you're well, and stop them if they stop working. And that can become quite a burden for some patients. Now, I said the logical place to start was in a disease where you weren't getting as high responses as you'd like to. We set the bar pretty high. We started with the disease where we saw a 79% response rate, chronic lymphocytic leukemia, and we asked the simple question, if you added in a different targeted drug, the monoclonal antibody rituximab, would you get a higher response rate and would people do better? And also, could you then stop all the treatment if they were in remission? And the answer to those questions is yes, yes, and yes, and I'll show you that now. But again, this study was initiated here in Melbourne, at Royal Melbourne, and Peter McCallum, as well as at centres in the United States. And what we were able to achieve was now an 86% response rate. And most impressively, now half of all the patients achieved this so-called complete response. Remember, it was 20% with Venetoclax alone. Furthermore, about a half of all patients, we couldn't detect any disease using our most sensitive tests, so there was no evidence of minimal residual disease. What's that meant is that people who've gone into remission have typically stayed in remission, and the length of time for that is on average well beyond 2 years, and we're still following many patients, so I don't have the final answer for that for you. But again, this seems a step forward. The other big step forward is that now in these patients who've achieved a complete response, in some cases we've opted to stop all the treatment and see whether they stay in remission. And that's what's shown here. The green bars show you the time on the Venetoclax drug, and the blue bars show you the time off all treatment and in remission, unless there's orange bars. And you'll see 2 people have orange bars. Both of those people, the disease came back 2 years after we stopped all treatment, and then they responded again when we gave them the Venetoclax again. The other patients remain in ongoing response of all treatment. And I think this is showing us the way of the future. With this targeted therapy, it's going to be one where we're able to combine it with other drugs, get a good response, get a remission, and then think about stopping. Now, we've also conducted trials in other diseases in combination. The one in CLL was the first. Other areas are in multiple myeloma, and again, we now see an increase. If you look along multiple myeloma, 21% response rate, Venetoclax alone, now in combination 67%. Complete responses were 6%, now 20%. Indirect, imperfect cross-trial comparisons, scientifically a little bit dubious, actually a lot dubious. Nevertheless, the general impression is that that looks like an improvement. In acute myeloid leukemia, again, about a third of the patients responding, now 3/4 of patients responding when it's combined with some other drugs. And now we're starting to see complete response rates in the order of 70%. And for acute myeloid leukemia, that's fantastic. Now, these are not final results. The trials are ongoing, so we need to watch this space. The drug is not licensed in any of these circumstances. So what have we learned from the combination studies to date? Well, firstly, that you can partner Venetoclax with different classes of anti-cancer agents that have already been proved to work. And I've shown you antibodies. I haven't shown you tyrosine kinase inhibitors, but an ongoing trial here in Parkville has demonstrated that. And there are trials with standard chemotherapy ongoing. I've shown you that you get augmented responses when you add other drugs, and you can do it safely so that the side effects are very manageable. And what's more, it looks particularly promising in diseases such as acute myeloid leukemia, and we'll watch that space. And we've already shown significant durability in CLL. Very excitingly, I think I've shown you that if you get a very deep response, complete remission, that it opens the possibility for stopping treatment, and I think that's a boon for patients. The final things to say are the bottom 3 dot points. Firstly, this drug should be paired with a partner that makes sense for the specific disease that you're addressing, so it's not a one-size-fits-all, and that needs to be driven by research. Secondly, I think we're just at the beginning of working out the potential of this class of drug in patients with cancer. And the final thing to say is that, remember I said that there are other proteins in the cell that work like BCL2. Well, where a BCL2 inhibitor we thought should work but doesn't, I think we need to rethink our strategy and say, actually, we probably need to target another protein in that cell that's keeping it alive, even though we've targeted BCL2. And with that in mind, there's ongoing research trying to address another protein in the cell called MCL1, shown here on the left-hand side of the slide. The details don't matter, but in some cells it will work like BCL2. And for some diseases, including some that I've shown you, acute myeloid leukemia and myeloma, we think it might be at least as important as BCL2 or more important. And again, so watch this space. So, since 1988, what have we been able to achieve? Well, that pivotal scientific discovery in Melbourne, followed by many other scientific discoveries in Melbourne, has delivered a new class of anti-cancer drug called the BH3 mimetics. Remember, they work like BH3-only proteins. A drug that's now licensed in multiple countries that specifically targets BCL2, and that drug's name is Venetoclax. The science done in the lab led to world-first trials being conducted here in Melbourne because pharma came down here because we knew what we were talking about. It's led to ongoing research both here in Melbourne, around Australia and around the world in how to best use this new class of drug, the BCL2 inhibitors, in many different blood cancers and some other cancers. My colleague Jeff Lindeman at WeHi and Peter Mack has explored this in some types of breast cancer, and it seems to have a potential role. And the number keeps going up, but there are more than 60 other trials ongoing around the world with this drug at the present time. Furthermore, it's fueled interest in this type of approach to developing drugs. And so we're trying to develop inhibitors of MCL1 and other proteins so we can target the Achilles heel of different cancers. I think all of this illustrates the power of collaboration. And the fundamental collaboration here was between US pharma and academia here at WeHi. But that was in developing the drug itself. But for that to happen, there had to be a lot of collaboration between different types of basic research: biologists, structural biologists, medicinal chemists, bioinformaticians. It had to be between multiple institutions and organisations, and it had to last over a period of what turned out to be decades. There was a lot of interaction between basic scientists and clinicians, particularly here on this campus in the form of clinician scientists, and then of course with people involved in clinical trials. And the results, I think, today are a testament to the quality of the clinical trials that we can do here in Melbourne. And ultimately then, it was a collaboration between pharma, hospitals, research institutes and patients, and some of them are highlighted here. On the right-hand side, there's a picture of David Wayne, who's on the right-hand side, who was the local leader in Melbourne driving the development of this drug, and 3 PhD students who all happen to be oncologists or hematologists that David and I supervised during their PhDs, and each of them made a major contribution here. And on the right-hand side, the trials wouldn't have been possible without outstanding research staff, some of the people shown here. Other outcomes, well, we had to attract pharma to come down to do this first-in-human study here, and subsequently there's been a lot more phase 1 clinical trials in the blood cancer space, not just from AbbVie and Genentech, but from other companies that have come on the back of this success. And what's more, it's attracted a significant amount of fundamental research funding and philanthropic funding to look for new ways to target cancer. So the whole thing is self-feeding. People like to back success and want to see new success arising on the back of previous success. And so I think we can hold this up as an exemplar for what can be achieved. Which finally brings me to the VCCC. I think the project that I've outlined for you is a perfect example of what we hope the VCCC will deliver. In part, the VCCC was involved at the end stages of the development of Venetoclax. But when we think about things, the potential of the Victorian Comprehensive Cancer Centre amongst all the alliance partners is truly outstanding. There's high quality basic research at many of the institutions that are part of the VCCC. Different complementary research expertises. We have the capability to conduct screens for new drugs, which are essential for the development of new anti-cancer drugs. We have a track record of collaboration and partnerships with industry, and that's growing every day. I've mentioned the very high quality clinical trials. They're only possible because of the very high quality clinical services. But high quality clinical services aren't sufficient. You actually have to have a robust clinical trial infrastructure. And I'm pleased to say that the VCCC has that in spades, and it continues to grow over time. So we have established track record in the clinical trials of new drugs, and Venetoclax is an example. So I'd just like to finish by saying that Melbourne has delivered a new drug for the care of cancer patients around the world. It's early days. The true impact of this medicine is not known. We know at the moment it's an option for people with tough CLL where other treatments no longer work. But I think the research says that it'll have much bigger impact in a wider range of patients with different types of particularly blood cancers. In finishing, I want to acknowledge a huge number of people who've contributed to this. At the Walter and Eliza Hall Institute, where it all began, more than 100 scientists over 20 years have put a lot of time and effort into the basic biology here. I want to call out David Wayne, who's been my partner in crime for the last 16 years since we embarked on this. And then below David, I've listed the 4 PhD students who have been integrally involved in the development of this drug. I mentioned Andrew Way, Kylie and Sion before. I want to call out Marianne Anderson, who was pivotally involved in the first clinical trial. My colleagues at the Royal Melbourne were instrumental to us getting to that point in the conduct of the trial, as were John Seymour, Contam and colleagues here at Peter Mac. And, you know, the wonderful thing about the VCCC is we're now one haematology service across both hospitals. This is where the trials started. This is where the discovery of activity occurred. The patients got great care because of the work of our research nurses coordinators. And we had a lot of people refer patients to us to be part of these trials. And again, that's part of the collaborative spirit of the VCCC. It was all done in collaboration with doctors in other parts of the world. Wouldn't have happened without pharma, AbbVie and Genentech. It absolutely wouldn't have happened without patients and their families prepared to trust us. And a lot of the basic research was driven by research funding from the organisations I've listed there. So I'll finish there.
Any questions for Andrew? Is there a facility in Melbourne to test for a particular cancer?
That is, so many pathology laboratories can test that. So for some cancers it's routinely done, for others it's not. I think there's already a wealth of knowledge about whether in a particular type of cancer BCL2 is highly expressed or not. So at this point in time, I don't believe there's the need for a special facility to specifically test that. One thing that I do want to point out, though, is while it's important that BCL2, central BCL2 is there, it's not sufficient. So we have examples of cancers that have too much BCL2, but this drug doesn't work. What that tells me is for the drug to work, you've got to have a lot of BCL2, but you need to know more about the cancer than that before you can think about having treatment with it. And that's a harder bar, and that's why we need clinical trials, and we need to go about things in a robust research framework to work out which people are likely to benefit from this drug.
Any more?
Yeah. So, yeah, thanks, Elizabeth. Not asked that question all that often, but it's a good question. And the reason it's a good question is because 79% actually probably underestimates the proportion of people with CLL who'd respond to this drug. Those trials were done in people with the worst of the worst CLL. I showed you we started with 2 higher doses, then we went to 2 lower doses. And so included in that 20-odd% of people who didn't respond were people who ultimately didn't get enough drug. And some of those people had side effects related to their CLL or related to the Venetoclax that meant they couldn't take it enough. And so I think if we did the trial again, knowing what we know now, we might get a higher result. But that's a supposition. The bigger problem is that we know some people become resistant to Venetoclax over time. I can't tell you why that is. It's not because BCL2 disappears, and we're still doing a lot of research on that. So maybe I can tell you in a year or two.
Everyone, I know you'll probably spend 2 hours to answer this, but how many other lecturers in Australia, let alone Parkville, we hear the same sort of story of another drug that we had basic science in the 1980s, 30 years of work, and then phase 1 study published in the New York Journal? How common would this sort of thing be?
Oh, it's pretty uncommon, Mark. As you say, it's not for me to say. Look, what I would say is that I grew up in Brisbane, and but I happened to come to Melbourne in 1992, and I came to Melbourne in 1992 because there has been a previous example, and that was the the development of a drug called GCSF, which is a supportive care drug for people having cancer chemotherapy. That was discovered in Melbourne, and the first trials were done in Melbourne, and the first in human study was published in a prestigious journal, not the New England Journal of Medicine, but the Lancet. And that story inspired me to what can be achieved when people collaborate together, and that was a collaboration between Professor Don Metcalf at the Walter and Eliza Hall Institute of Medical Service, George Morstan at the Ludwig Institute, and Dick Fox and colleagues at the Royal Melbourne Hospital. So I've seen it once before. I think the thing about the VCCC is I'm hoping to see it multiple times before I retire. We have the opportunity with the collaborations we have within the VCCC to do this again and again.
On that note, and that very optimistic note, I think my instructions are I've got to remind you that you should please register for upcoming presentations that include Dr. Dishan Harath on artificial intelligence for cancer diagnosis and therapy, Dr. Mark Dawson on the cancer epigenome, and Associate Professor Jeff Pulse on targeting nutrient uptake to starve cancer cells. So there's an ongoing list of absolutely wonderful presentations that will be taking place right here this time following Monday. So please come along. And lastly, let's please thank Andrew for an absolutely wonderful